Hello students. So today we'll be doing about the elongation of DNA replication. Okay. Elongation of DNA replication. As I had discussed in the last video that this is the replicating fork, all right, which goes from 5' to 3', 3' to 5'. All right. Now there is another enzyme. Just a quick revision about what we have done in the last video in the initiation process. So hexamo is there, helicase, right, with DNAC. Okay. So what happens, there is another enzyme called the DNA polymerase 3. The DNA polymerase 3, which will the DNA pol 3. Okay. DNA pol 3, which means DNA polymerase 3, it is the main enzyme. The main enzyme that takes part in the elongation of DNA replication, or in the whole process. The DNA pol 3 is the most important enzyme because it takes part in the polymerization of the DNA strand. Polymerization, adding of new nucleotides, isn't it? If we add the new nucleotides, what will happen? A new strand will be formed. All right. So a DNA polymerase has a problem that it only acts from 5' to 3' end. That means it will only run from 5' to 3' direction. Okay. Now if the parent strand is from 5' to 3', then the DNA polymerase will whatever the chain, the next chain, the replica that it is going to make, it will make from 5' to 3' end, right? That is the main direction that a DNA polymerase 3 follows. That is 5' to 3'. It always work in the direction 5' to 3'. So here what happens, before the DNA polymerase 3 is uploaded, there is another enzyme called the RNA polymerase, which is altogether known as primer. A primer is uploaded before this. Okay. This is the main enzyme that helps in polymerization, but before this, another enzyme, that is a primer, is added. A primer is added. All right, that is a RNA polymerase. Okay, and RNA, you can write it in. RNA polymerase. A primer is uploaded first here, but RNA polymerase makes only 15 or 10 to 20 nucleotide chains. All right, that will not help, right? We have to add number of nucleotides so that the polymerization takes place and a new DNA will be formed, DNA strand will be formed. So RNA pol, and that too RNA pol will be adding the RNA nucleotides, not the DNA nucleotides. But in the process, the primer is uploaded and makes the 15 to 20 bonds. Okay, 15 to 20 nucleotides RNA nucleotides will be added, okay, by the enzyme RNA polymerase, that is also known as a primer. A RNA polymerase, that is a primer, is uploaded here, okay, in the new direction, in the new strand, which is from a 5 prime, okay. This is a 5 prime end, and RNA polymerase will form the RNA nucleotides, okay, while the nucleotide that should be added, that we need to make a new strand, is a RNA nucleotide, DNA nucleotides, not RNA nucleotides. But RNA polymerase is there, so that's why it will add its nucleotides, that is the RNA nucleotides. Now what happens, the DNA polymerase 3 is uploaded now. Since for DNA polymerase 3 the way is very clear in the uppermost, in the upper strand, right, because as it works, DNA polymerase 3 works from 5' to 3'. In this, the upper strand, what is the direction that a DNA polymerase is getting? 5 prime to the 3 prime direction, right? 5 prime to the 3 prime direction. So what happens? It will be continuously keep on adding the new nucleotides, the DNA nucleotides. Now here you will get confused like this. So this is the RNA polymerase, okay, which is adding 10 to 15 nucleotides. All right. While now a DNA polymerase 3 is added, and it makes a continuous strand in the upper parent strand of the DNA. All right. The replica that it is forming is from 5 prime to 3 prime end, okay, which is the main direction of DNA polymerase 3. So it will keep on adding the nucleotides and keep on making the new strand. While if you see this direction is 5 prime to 3 prime, then the down direction, let us make it again, okay. All right, this should be 3 prime to 5 prime, right? Now the replica, the daughter DNA strand that is produced, will be here from this direction, right? Now it will be from this direction, right? But DNA polymerase is only one, right? In the procedure, the DNA polymerase is only one enzyme, but it has to work in both the strands. It has to add the nucleotides in both the strands simultaneously. But how is it going to take? How is it going to work in both the strands? All right, because in the upper strand, what is it doing? It is moving in this direction, right? Because it is the main direction that a DNA polymerase 3 takes, 5 prime to 3 prime. But since this way is opposite to the direction that a DNA polymerase 3 takes, how come the DNA polymerase can work in both opposite direction? If it is here, if it is making the nucleotides here, it cannot just simply jump and can make the nucleotide from this place, right? So what happens? There is a loop that will be formed from 5 prime end. This, the parent downward strand of parent DNA strand will form a loop, and this 5 prime will come here. All right. If it forms a loop, it has made a direction here. Okay, of course, when the DNA polymerase is working and making the continuous strand in the upper one, this will be the lagging strand. Why? Because this is the continuous strand that a DNA polymerase is making and going on. But in the second strand, what is happening? In the second strand, since the orientation is in opposite direction, there is a loop that forms here, and 5 prime end comes here. And for example, a DNA polymerase is making the chain over here, going over this position. What happens? In the downward direction, a loop is formed and 5 prime comes this side. Okay, the upper side, so that the polymerase can act now in, of course, the helicase is acting in both the strands at one time, and then opening up the strands, because the nitrogenous bases are present like this. Okay, they are joined here, still joined, right? They are still joined, right? So DNA B, the DNA B enzyme, that is a helicase, is opening up this. All right, and DNA polymerase 3 is acting on this side, while since the DNA polymerase 3 cannot act in opposite direction, cannot go in opposite direction, one enzyme cannot work in opposite direction at a time, what happens? The downward strand will form a loop like a structure, and then it will come a little forward. Okay, it will come a little forward, so that the DNA polymerase, it will be easier for DNA polymerase to act in both the strands simultaneously. All right, only the problem here arises is that the DNA polymerase now will at a time will act in this direction, making it a leading strand. And since the RNA primer here was first added, what happens? What happens when the DNA polymerase 3 is acting over this strand? The RNA primer will come and make the bond and make or add the nucleotides here in this strand. Okay, but it will make in the fragments. Right, now what happens here? Again, simultaneously, this DNA polymerase 3 will come and act in this direction, that is 5 prime to 3 prime. Since it has made the loop, it has come little forward, and it can act. DNA polymerase can add its nucleotide in both the strands simultaneously, but the direction of the adding nucleotides will be opposite. Okay, it will work simultaneously but in opposite direction. All right, since it is one molecule, it cannot work without changing its orientation. All right, it has to work from where it is doing the replication. It is adding the nucleotide in the first strand. It should in the same position or in the same way, it should make it should add the nucleotide in the second strand also, but it will make it in the opposite direction. It will go in the opposite direction. Okay, and that too in the fragments. will add the nucleotides in the second strand in the fragments-like structure, which is known as the Okazaki fragments. All right, and then this is the work that a DNA polymerase 3 does, that is the polymerization of DNA strand, and due to which the new replicas, the daughter replicas of DNA is formed in both the strands, in both the parent strands. Okay, and this is a lagging, since it is making it is adding the nucleotides and fragments, it is known as the lagging strand. All right, so this is the leading strand, this is the lagging strand. Okay, leading strand, this one, and this one is the lagging strand. All right, and it will be having the direction from 5' to 3' end, that is this direction from 5' to 3', and this direction from opposite direction. Okay, here also only one enzyme, that is a DNA polymerase 3, is acting in both the strands, but since the orientation is different, the direction is different for adding the nucleotides in both the strands, and since the RNA primer adds the nucleotides in the fragments, the DNA polymerase 3, when comes to the lagging strand, it finds little space and adds the DNA nucleotides in the fragments-like structure known as the Okazaki fragments. That is why the second strand is known as the lagging strand. All right, and now another enzyme called, since it is a RNA polymerase, that is a primer, is adding the RNA nucleotides. These are the RNA nucleotides, right? Nucleotides. So we have to remove it. Remove RNA nucleotides because it is the DNA chain, DNA polymerization. So the DNA nucleotides should be added. So for that particular process, for the removal of RNA nucleotides, we have another enzyme called the DNA polymerase 1, which has 5' to 3' exonuclease activity. Which shows the 5' to 3' exonuclease activity and helps in the removal of RNA nucleotides. All right, and then since it will remove the nucleotides from here, right? For example, if this is a DNA nucleotide and this is the RNA nucleotides, okay, RNA nucleotides added by the primer and DNA nucleotides added by the DNA pol 3 or lagging strand. Okay, I'm just showing the lagging strand because in the leading strand there is no problem like this, because DNA polymerase gets it a proper direction from 5' to 3' and makes a continuous strand in the leading strand. While in the lagging strand, since number of RNA nucleotide fragments are present, we have to remove this. While in the leading strand also, there is one place, the primary enzyme that acted in the polymerization of DNA chain is a RNA polymerase. So to remove these RNA nucleotides, the DNA polymerase 1 acts and removes this RNA nucleotides, okay, by the action of DNA pol 1. Removal of RNA nucleotides takes place and it adds the DNA nucleotides again, and this is how the another replica, the new replica of DNA, a daughter DNA, is produced. A daughter DNA strand is produced. But since DNA polymerase 1 can do mistakes can happen anywhere. To see if the DNA polymerase 1 has done any mistake or added any type of wrong nucleotides, DNA nucleotides here in the lagging strand or the leading strand in the process of removal of RNA nucleotides, what happens? There is another enzyme called DNA ligase, which comes and binds to the strand and see that, okay, this DNA ligase comes here and binds here and see that there is no mistake that is done to form to add the nucleotides, okay, DNA nucleotides. So it does the proofreading, all right, proofreading of the DNA nucleotides chain. This is how the DNA strand, the parent DNA strand, these two strands, the parent DNA strand will form the new nucleotides. So now these RNA nucleotides are removed and the DNA nucleotides are added, and so the two new daughter DNAs are formed, right? These two are formed, two strands, right? This is what the two daughter strands will be formed by the process of polymerization chain reaction, all right? This is how the full process of DNA replication takes place. Now I will be showing you about the whole process. I'll just give a overview about the process. So in elongation, what happens? RNA polymerase is added and adds the RNA nucleotides, and then DNA polymerase 3 is added, which always works in the 5' to 3' direction. For leading strand there is no problem. For lagging strand, since the orientation is different, since there is only one enzyme, that is the DNA polymerase 3, it to act in both the strands simultaneously, in the first and second parent strands, what it does, it forms a loop like structure, and then the orientation of it is changed, and then the polymerase 3 will act, adding the nucleotides in both the strands but in different directions, okay? The lagging strand is formed and leading strand is formed. In lagging strand, fragments of DNA will be RNA nucleotides will be added, and in the space left, the DNA polymerase 3 will be adding the DNA nucleotides, and by the help of DNA polymerase 1, which shows 5' to 3' exonuclease activity, will remove the RNA nucleotides, and then DNA ligase will see that there is no mistake done in the process of polymerization process where the DNA nucleotides are added. Okay, this is how the two replicas of DNA is formed. Two daughter DNA are made from a parent DNA. So thank you for today. This is what is the elongation process of DNA replication. Now in next video I'll be teaching you about the termination point. Thank you all of you.